Welcome to the second lecture of this course, and today we will work on DNA analysis, which is one of the core skills that you have to develop as a biologist Pythonista. And it's also a great data type to play around with Python and to learn some basic Python skills. So what we will learn today will be working with strings. DNA can be represented as string data in Python. Um, we'll, we'll build some tools that are typical for DNA sequence analysis. And this involves a lot of pattern recognition kind of algorithms. So that's what we're doing today. The skills you learn is dealing with dictionaries, which is another container type where you can store information and data in a more structured way. We learn how to deal with strings, following up from last lesson. And very importantly, with every programming language, we will get into conditional logic and make bifurcations in our program. If something happens, we'll do this. If it doesn't happen, we will do that. This is a very important element of computer programs. Additional topics we'll cover today is we'll dive a little bit at the end into further resources and start talking about open source software and especially BioPython. And I won't do this in this lecture, but in the labs we'll start talking about Python development tools and setting up your own Python environments. And I will work on the resources and update the videos in the resources as well. Right, so the goal today will be to read in a DNA sequence and then analyze it and find the open reading frame. And I will, for those of you who don't know what open reading frames are, I will explain that. So it's finding the start codon and then finding the stop codon, extracting the ORF, and then translate this to the protein. We'll leave it at that, but we could also then find all the open reading frames in the sequence and just get the longest one, for example, and we could make that into a loop thing to process many files at once, but the core program will be what we will focus on today. And then if you want to, you can develop this further. And to start off with, always there's a problem. This time it's Darren, a postdoc, and he is pretty frustrated because he's been given hundreds of sequence files and he has to messenger RNA files and he has to find the open reading frames. So this is the starting point, and this is a typical condition that you'll find yourself in a lab where you have to get a lot of data, you have to analyze them quickly. To do this manually is very tedious. There's some good programs to do it one by one, but if you have a lot of this data, even with a good program, if you have to do it manually, click by click, it'll take you ages. And with the programming approach, you can cut this down into a very, very smart solution. Again, automation and Python. So a brief detour just to get the biology right. So every messenger RNA will be scanned by ribosomes for an ATG. And when it finds the ATG, it will start translating this into proteins. And then ATG itself is a codon. So codons are always triplets of bases that stand for one amino acid. So ATG stands for methionine. It will then read in steps of 3. So this is the genetic code. It will add the amino acids that these codons encode until it finds a stop codon. In this case, it's TAG, and then it will fall off and stop producing. So we want to find the first ATG, then read in triplicates the individual codons until we find TAG, and that will be our open reading frame, and we will then translate that into amino acid sequence. Very nice biology problem, and also very nice coding problem. So the key concepts here are that DNA can be read in different frames. So there could be also an ATG starting here, for example. But we just want to keep it simple in our program. We just want to find the first ATG. And in terms of coding, again, as last time, always start with breaking down the problems into smaller problems and then deal with them one at a time and then put them together into a final complete product. Remember the Lego comparison. It's really like that you build little Lego blocks and then you put them all together. And here functions help us very much. The thinking in function is really helpful. You can see what are the individual steps, and then you build functions around them, and then you combine that into a main function that brings all the other functions together. So that's what we'll do today. And the three steps that we have to do is we have to find the first ATG and then read in steps of 3 until we hit a stop codon and extract that information, and then translate these individual codons into messenger RNA and build the protein sequence. So what we'll deal with here is string slicing, loops and logic and conditionals, dictionaries, and then in the end we'll have a brief session around debugging and fixing errors and dealing with errors. This is a very important part of this lecture. And to finish off this brief introduction here, I'll show you the complete code. So this looks quite advanced already, but not to worry. Most of these things you already understand, except for the conditionals, which we'll cover, and the dictionaries. But let's run this. So we have several functions: find the ATG, find the ORF, translate the ORF, and then combine everything into a main function that we call find protein sequence. And when we run this function on a sample DNA sequence, you see we get a nice result that we can then print out as our searched for protein sequence with a little star for the stop codon. So this is what we're going to deal with. So what we have to learn is what are these weird things in curly brackets, and how do these if statements work, and how do we pop along strings in this manner to find stuff. So those are the skills that we'll cover today.